Hey everyone, welcome to CAPSA. In today's session, we will understand about vesicular monoamine transporters, also known as VMAT. These transporters play a key role in the storage and release of neurotransmitters like dopamine, serotonin, and norepinephrine. So, what exactly will we cover in this video? So, first we will understand what VMAT is and why it's crucial in neurotransmission. Then we will explore the two types of VMAT, 1 and 2, along with their distribution in the body. And finally, we will break down the structural features of VMAT proteins and their genes encoding the mechanism. So let's get started. Let's begin with the basic question. What is VMAT? Vesicular monoamine transporters are specialized proteins embedded in the membrane of synaptic vesicles. Their primary role or the job is to transport monoamines, which include dopamine, norepinephrine, serotonin, epinephrine, and histamine, from the cytosol into the storage vesicles. Now, why is this transport so important? Well, if these monoamines remain in the cytosol, they get rapidly degraded by monoamine oxidase or MAO. So VMAT protects these neurotransmitters by sequestering them inside the vesicles, where they are safe, stable, and ready for release during the neurotransmission. And importantly, VMAT uses a proton gradient established by vacuolar type ATPase, a proton pump that maintains an acidic interior in the vesicles to drive this active transport. Now let's differentiate the two types of VMAT, 1 and 2. So VMAT1 encoded by the SLC solute carrier gene 18A1 gene, which is primarily found in the peripheral nervous system, and you will see it mostly in the chromaffin granules of the adrenal medulla and enterochromaffin cells of the gut. Its main function is to transport norepinephrine, epinephrine, and serotonin. On the other hand, VMAT2 encoded by SLC18A2 gene, which is dominant transporter in the central nervous system. It is highly expressed in dopaminergic, serotonergic, and noradrenergic neurons in the brain and spinal cord. In addition to other monoamines, VMAT2 is also responsible for histamine transport in the brain. So, to sum it up, VMAT1 works mainly outside the brain, while VMAT2 is the critical transporter inside the brain. Let's now look at the structural features of VMAT proteins. Both VMAT1 and 2 are integral membrane proteins with 12 transmembrane alpha helices. These helices traverse the vesicle membrane and help form a central cavity, the site where monoamines bind during the transport. The N-terminal and C-terminal domains of the protein are located in the cytoplasm. These regions often carry regulatory signals such as phosphorylation sites, which can influence transporter function or trafficking. VMAT works using a proton antiporter mechanism. This means it exchanges protons or H plus ions for monoamines, essentially using the acidic environment inside the vesicle as an energy source to pull the neurotransmitter inside. There are also glycosylation sites on the extracellular loops, which can impact stability and trafficking of the transporter to the vesicle membrane. Let's quickly recap what we have learned today. So VMATs are essential for the safe storage of monoamines inside the synaptic vesicles. VMAT1 is mostly peripheral, whereas VMAT2 is central, both working through a proton gradient using energy to concentrate neurotransmitters where they can can be released on the demand. Structurally, they share a common design with 12 transmembrane domains and complex regulation through phosphorylation and glycosylation. So in the next video, we will explore exactly how these transporters works, including the ion exchange mechanism, proton coupling, and what happens when this system fails, and how it's targeted by the drugs. So if you found this lecture helpful, make sure to subscribe to CAPSA so you are notified when our next video on mechanism of action of VMAT goes live. If you have questions, drop them in the comments below. Thanks for watching. See you in the next class.